So let's start. We are done with the exam, and you know that probably you just do have some questions and all those kind of things. We're just holding on to those. There are just a couple of very legit makeups that needs to be given, and I will talk about that. And what I will do is, like, as soon as the makeups are done, I will open up the exams, so you will be able to see the correct answers. You will be able to see that what is your response, and you can, of course, after that, you can, whatever question you have, as you do, you email that if there is any problem with any questions, and at that time I will just address the same way. So it will be completely transparent the same way for the fourth exam as well. So you guys are almost done. So last module, and what you're going to see that the first part of the module, which is 4.1, is 2 parts. So approximately we have about in between 28 to 30 slides for these 2 parts. And you may think that it is a kind of a lot, but when we go through this material, you will be able to see that a lot of terms that we are using here, you already know those and you have a basic understanding. So it will just really be helpful for you guys. And I will just give you some pointers that there are some memorization type of material that is needed. But again, you know that you can have a printout of that and keep that handy. So that will again help. And another thing is that I know that I can just use the Respondus monitor, which will again going to record you and all those kind of things. I'm just not using this at this point. It just gives you, probably it's rigorous enough, and just a little bit of help that you can use your notes or books if it's needed. There's not enough time that if you do not study, then you cannot look it up within that 75 minutes, but that can just help a little bit. On the other hand, the next part, which will be the 4.2, there will be 3 parts, which will be the cancer biology, and altogether, in all 3 parts altogether, you will have 50 slides. So pretty much you are just getting approximately 60 here, 50. So that will be a total number of slides. And we probably will have 1 extra lecture day that we are probably going to talk about just before the exam, the last Thursday, just only review of the exam. So that's about it. So let's start with the material. So as you all know that the cells do not live in isolation. Any questions I did not ask? Any questions, anyone so far? So I guess everybody good. Okay. So the main thing that what we are looking at, the cells do not live in isolation unless it's an eukaryote and it's a protozoa. I mean, like protozoa onwards, everything is multicellular. So there is interaction in between the cells. So in eukaryotes, particularly for us, for the human beings and other mammals, other higher animals, the cells altogether give rise to the tissue. So similar type of cells, and you already know from the previous chapter that we have approximately about 200 different types of cells in our body. And not all of these different cells give rise to that many tissues. A single tissue will have cells which are related in function, but a single tissue can have multiple types of cell. Say, for an example, your skin or connective tissue, a particular tissue may have, say, I'm just taking blood. Blood is a connective tissue. It's a liquid connective tissue. This is not needed for your exam, but you have so many different types of cells. They are all related, but they are not the same cells. So they interact in between each other. And also another important thing is that surrounding the cell, you have a space, which so far we called an extracellular space. And this extracellular space is just not empty. It is filled with many different types of fibers, protein, and many different types of proteoglycans, but mainly proteins and proteoglycans. Proteoglycans are usually protein chains where you have attached carbohydrates. Very important for your extracellular matrix. And all these cells are in this extracellular matrix close to each other. The difference is that in between the tissues, when the tissues are formed, there are some tissues where the cells are packed against each other, and almost there is no extracellular space, or the extracellular space is extremely small. And this is very true for your epithelial cells, say, which is lining your your skin or your intestine or your blood vessels. In between the cell, you have very little amount of extracellular matrix. Whereas in a connective tissue, you have abundant amount of extracellular matrix where you have these fibroblast adipocytes, which are fat cells. Fibroblasts make collagen fibers, different types of fibers. They are dispersed around the extracellular matrix. So there are some tissues where you have a lot of extracellular matrix, some tissues where you have less extracellular matrix. But no matter what, excuse me, but no matter what, all the cells interact with each other, and all these cells will interact with this environment, which is extracellular space as well. And that's what the focus of this chapter is. And once you have a tissue, a multiple tissue will form an organ. And that's the important part. So you can see that, think about this, that you have the integument. I mean, like, there are many different types, but integument, your skin is an organ. But you have so many different types of cell. You have the keratinocytes, but there are other cells where you have some of the secreting cells, which do secrete some of the sebaceous secretions. Those are in there. There are some sentry cells of the immune system which wander around. There are some macrophages which are deeper into the skin. And then you have a connective tissue where there are many different types of the dermis part of your skin, where there are many different types of cells are there. But again, just the idea that many different tissues will form an organ. And here you can see just a typical tissue where you have the dark parts are the cells, and you can see the nucleus in very dark, that pointy structure. And all the white parts that you are seeing in between the cells are the extracellular matrix. So we are going to talk about two different interactions. One is a cell-to-cell interactions, how they interact. And another part is that cell matrix interaction. Now, remember that when you're talking about the cell matrix interaction, this interaction could be with a protein fiber, or it could be a kind of a sheet made by a protein, like a basement membrane. So that is what we mean by a cell matrix interactions. So let's take a look how we are going to do that. Cell-to-cell interaction as well as matrix, we can all see these through either electron microscopy, and even if we just do have specific antibodies to these structures, we can add a label to our fluorescence label to that antibody or the secondary antibody, and we can visualize these structures as well. So these are some of the ways that we can visualize those. All right, so this gives you an overview of that, and this will make you a little bit feel better. That the first part is that what we're going to talk about is the cell junctions, how these cells are connected to something else. The first one is occluding junctions. It occlude or doesn't allow something to pass these junctions. That's why they are called occluding junctions. Example, tight junctions. Think about that. Your transmembrane protein lecture, go back to that. We have already talked about that. Then we have these anchoring junctions. And the anchoring junctions, if they are made up of actin, they are called adherence junction. And we have already talked about the adhesion belts, your actin lectures, and also the cell matrix, which is called the focal contacts, and these are also actin lectures. If you remember that cell to matrix actin interaction is that if you remember that on a state matrix, there was a cell which is extending its lamellipodium and moving from left to right, that was your focal contact cell matrix. And you will see that we will use the same pictures later. So these are all familiar to you. We will go a little bit more detail. These are the ones which are new, and we are introducing these. If you have anchoring junctions with intermediate filaments, cell-to-cell interaction is called a desmosome. So if two cells are stitched together, they are using intermediate filaments, they will be called desmosomes. On the other hand, if it's a cell matrix interaction, it's a hemidesmosome. And most of the times, if you remember, that your epithelial cells rest on a basement membrane. And usually these hemidesmosomes will keep these cells attached to the basement membrane. So that is a cell matrix interaction, and your basement membrane is a matrix, extracellular matrix. And we'll see that. And the last thing that we're going to talk about is the communicating junctions, which is a gap junctions. And we talked about that just briefly, just as an example. in cell signaling, where we talked about that small molecules can pass through these for signaling purposes. So that's basically the overview of that. And these are the only two terms that you are seeing being introduced for the first time. The rest, you have at least you have an overview of idea that what they are. So let's start with that. We're going to talk about each one of them. So right here, we are looking at just a cell, and to think about that this is just a single layer of epithelial cells, which is of course polarized. So you can see that this is your apical surface. Right here, you have a basement membrane, which is known as the basal lamina. And that is your extracellular matrix. And here you can see that this whole part, even below the membrane, you have this connective tissue with lots of these fibers. So your basal lamina, which is the basement membrane, as well as this part of the connective tissue, which is a loose connective tissue. And again, you do not have to know the detailed name of any of these connective tissues, but all together they form the extracellular matrix. And the cell, particularly in this type of cell, you can think about if it is a typical epithelial cells, you have an apical surface, and you have a basal surface and a lateral surface, which also we call as a basolateral surface. And the basal surface is stitched to this basal lamina or the basement membrane. Both are the same thing. So let's take an overview of that. What are the different types of junctions that is present? The first thing that what you are seeing right here is a junction, which is a tight junction. You can easily see that this is a tight, this is a tight junction, so it's right here, and we're going to talk about that. Just below that, both sides, you can see just below the tight junctions, and usually it is strategically placed, these junctions, below these tight junctions are what is called is adherence junctions. Adherence junctions, and these are made up of these actin filaments which are radiating out from there, and we are going to talk about detail about these actin filaments. Adherence junction made by actin filaments. Then what we are seeing that another two structures in between the two adjacent cells, there is another structure which is the desmosome. Looks very similar, but see the color of the filament is different, which tells you that this desmosome filament is an intermediate filament, and we are going to talk about that detail. So here it is actin in adherence junction, desmosome, it is an intermediate filament. Could be many different types. Then you have something which is called a hemidesmosomes or hemidesmosomes. These anchor the cells on the basement membrane. And the reason for that, think about that, if there is food material passing through here, constant mechanical action will just take these cells off and move along with that, and they will, although that happens all the time, and that's the reason that these cells need to be renewed from your stem cells. As you know that they are constantly making these cells, they are replaced every 3 to 5 days because they do come off. But if you don't have these hemidesmosomes and these cells anchored onto the basement membrane, they will just be these cells will get teared off from the basement membrane or the basal lamina all the time. And then you have another, the last one is the gap junctions. What we are going to see that these actually form pores or connections in between the two adjacent cells. And these pores are very, very small, very selective as well. And these are called the gap junctions for signaling in between the cells. So these are the overview of that. The only thing that is not shown in this is the focal contacts, which we are going to talk about later, which is also the cells, how they attach to the matrix. All right, so here you can see that this is your biomembrane directly from your biomembrane lectures, and we are just going to spend just 30 seconds on this slide. So the first thing of these occluding junctions are the tight junctions. And what they do, they maintain the polarity of the epithelial cells. And you know that this is the basal lamina. So this part, the side and the bottom, is the basolateral surface. Whereas right here, the top part is the apical surface. And what you are seeing that you have a protein A, which is green on the apical plasma membrane, whereas you have this red protein, protein B on the basolateral surface, but they are polarized, two different proteins. And who maintains that? Right here, the tight junctions. These tight junctions, and that's the reason we call this as a occluding junctions, these doesn't allow the proteins to move in the basolateral part, nor it allows these proteins or the protein Bs right here to move on the apical part. And always remember that, just try to visualize that we are looking at always these two-dimensional structures. But remember that all these cells are in the three-dimensional structure. So this particular tight junctions, they run around the periphery of the whole cell. It is not just on one area. So sometimes it is very difficult to kind of visualize that how this actually works. But it is 360 degrees around the cell if the cell is round at this point. So that's your tight junctions. So that's the overview of the idea to maintain the polarity of the cell. Different proteins needed, different transporters here, different transporters here. I mean, like, just to give an example that what are the different proteins. So very straightforward. So here are some examples that how you can study. You can put and how these tight junctions maintain this polarity. You can put a tracer molecule, which could be a fluorescent molecule. And if you just in real time look at them under the fluorescence microscope, you can see that if you just put a fluorescent tracer molecule in the apical surface, they will not be able to diffuse into the basolateral surface. So that's one of the experiments. So here you're seeing a similar kind of experiment with electron micrograph. What you have, that you have an electron dense material which is put on the apical surface of that. And right here is the intracellular junction. So what do you mean by the intracellular junctions? This is left-hand side. This is one cell. This is another cell. These are the microvilli which are projecting out of the cell. And these apical surface is with this dense black electron dense dye. And right here, you have a tight junctions followed by this basolateral surface and the empty space or in between the two cells, extracellular space, but this is the tight junctions. And this dye will not pass through this. That's why this is clear. On the other hand, this is from the other side. You are seeing that right here is a cell on the left-hand side, cell A, right-hand side, cell B. You can see the microvilli. So the top portion is the apical surface. Basal surface is not shown. This is basolateral surface, but what you are seeing that another electron dense dye, when put on the basolateral side, this cannot pass these tight junctions. So that's the reason that you can easily conclude that how the apical and the basal surface are occluded by these tight junctions and what's the function of it. Different proteins in different area for different functions. Straightforward. And they serve as the barrier for the solute diffusion. Very, very important, because if you just allow your basolateral surface right here, the solutes or the different ions and all those, say, amino acids or sugar molecules to pass through these areas, it will be completely a disaster. And we are going to, of the homeostasis of the cell, and we're going to talk about that how it works. So the next one right here, just an extension of that, and just tells you that what this is. This is a lanthanum hydroxide. Lanthanide is this is a very high molecular weight material. That's why it is very electron dense. And you can see that right here on the basolateral surface or the lateral surface of the cell, you have this particular electron dense material that cannot pass through the tight junctions onto the apical surface. Just an extension from the last slide. All right, so right here, this is also familiar to you in a almost similar way. You will see that. So what are the different ways that you have these different things, and are there any exceptions? So what you are seeing that if you have a cell which does have an apical surface right here, and you have the basolateral membrane or the basolateral surface. And as we said that these tight junctions separate the apical and the basolateral surface. So if you need transportation on molecule, most of the molecule are transported by this pathway, which is a transcellular pathway. The material from here will move in here and move there, or material from here, move in here, move there. This is a normal way, but there are few exceptions. And this is your paracellular pathway, where some of the molecules are allowed to move through the tight junctions. So it is not 100%, but this paracellular pathway does allow some molecules. But the important thing, that is your take-home message, that these examples are very, very rare. very rare. 99% of your material is moved through the transcellular pathway, less than 1% of the material moved through the paracellular pathway. So we are going to talk about that and some of the thing that you already know. So we are going to take a look at this. So if this is a familiar slide with you. This is your CFTR, whereas right here in a normal lung, this is your basal area. This is your airway lumen. And you can see right here, you have a CFTR. This is the channel transporter, which will move the chloride ion into the airway lumen. And that's the function of that. Again, remember that this is not a pump, but this is a channel because it is moving the chloride, which is high here, to down the concentration gradient in here. So once chloride is moved through the CFTR into the airway lumen, what it will do that due to this minus charge, it will again attract the sodium. So right here, you can see that this tight junctions will allow sodium to move into this airway lumen. So sodium is one of these exceptions which is allowed to move in certain tissues to move them through the paracellular space. And you can see that the water, it will not allow the water to move to the paracellular space. So water needs to be moved by the aquaporin inside the cell. And then again with the aquaporin, it will be able to come through this. There will be no water movement through the paracellular space. Only sodium can move through this. So what happens in a CF lung when there is an absence or defective CFTR, all the chlorides are occluded inside the cell. They will not be able to move in. No sodium will be pulled into this area. No water will be pulled into the airway lumen, which will cause a very thick mucus. So this is an example that you have seen. This is also another example that you have seen. And see, this is the consequence of the tight junctions, or why this tight junction is so important. That's what we are trying to understand here. I am not going to ask any of these particular pumps or how they function, what they do, nothing those. Those are just for an example that how in this context tight junctions is important, because we have already covered this. So what is happening here? So you're seeing that right here, you have your epithelial cells, suppose your intestinal epithelial cells. The left-hand side is the basal side, and this is right here is your basolateral side. Right here, right-hand side is the apical side. So this is compared to these cells, which are the top is the apical and the bottom is the basolateral side. These cells are sideways, not top-bottom. So here what is happening, that glucose from the intestine with the help of sodium is moving into the cell. And the glucose concentration is going to get high here. And you also know that the glucose is not readily going to get converted into glucose 6-phosphate here yet, because that glucose 6-phosphate, what you learned earlier, that's going to get converted into the cell where glucose will be delivered. Here, it is not going to get converted into glucose. Your glucose concentration is getting higher here. So what is next happening? Here you have a GLU2, which is basically making a glucose gradient. This is a facilitated diffusion. Remember that this is not a pump, facilitated diffusion. So this GLU2 transporter, which is only on the basal side of this cell, will move the high concentration of glucose from a region of high to the region of low. So it will just glucose coming inside the cell, glucose concentration is high and it is moving. The glucose out into the bloodstream where it will be delivered to proper places, mainly in the liver, and where it will be converted into the glycogen, or in the muscle where it will be converted into the glycogen. The sodium is maintained by a sodium-potassium pump, but these are the things which is going on. But why this tight junction is important? So what will happen is that, think about that suppose you have a complete connection of this basolateral surface as well as the outside surface completely. So then at that point of time, you do not have a gradient of glucose, but think about that. This is completely connected with the other part. There is no tight junctions. So this is continuous with this area. So that means that all the glucose which is in here, they will just readily pass in there, and there is no high concentration of glucose here, high concentration of glucose here, high concentration of glucose here, and it will be a completely disbalance, and your cells in your body will not be able to receive the glucose. A lot of these glucose are going to get wasted in that process, completely mess up the homeostasis. But see that what the tight junctions are doing, they are keeping the glucose in this intestinal lumen separate from the basolateral surface where it is going to get delivered into the blood. And what it is doing in the middle portion, it is taking the glucose from low concentration and making it a higher concentration. Remember that glucose is low here. And as it's constantly making the glucose come in here, it is getting higher, which is done by the sodium, because this is not a pump, direct, no ATP is involved, but as sodium is moving down the concentration gradient, it pumping the glucose from outside to here. So as the glucose concentration is high here, it will move the glucose from the cell here. If you don't do that and allow the glucose to completely maintain in that condition, see that what can happen. When you don't have a meal and you don't have any glucose in here, you still may have some glucose. This transporter, which is helps in the facilitated diffusion, will probably move glucose from here to inside the cell. So instead of going in this direction, it will completely mess up the gradient and move glucose in that direction. So that's the reason that why tight junctions or these occluding junctions are so important. All right, so let's move on to the structure of the tight junctions. So the tight junctions, what you are seeing, that suppose this is one of your cell, another cell, and the plasma membranes are separated. And this is your intercellular space in between, that what we were seeing, that suppose this is your space that you are seeing right here. So how the tight junctions work, you can easily see that they are stitched like this. And there are 3 proteins, that is the important part. And what are these proteins? There's a protein called occludin with 4 transmembrane domains. Then you have a claudin, which is also 4 transmembrane domains. And then is a JAM, which is just a single pass protein, transmembrane protein. And all these basically stitch them together. And this is the structure of a typical tight junction. So what you are seeing, that there are some parallel proteins which are running. Remember that they're running across the plasma membrane, 360 degrees around the cell. So this is your horizontal proteins right here. And they are also connected in between these by these proteins as well. So you have these, the attachment of these vertical portions along with that as well. And here you can see that how the tight junctions are. So this is basically the proteins that how the tight junctions are responsible for separating the apical and the basolateral surface. So right here, basolateral surface, top portion, apical surface. And just remember the name of the proteins which is involved in that. That's it. All right, first clicker question. Let's do it. So first, which of the following type of junctions are not found on the lateral surface of the epithelial cells? Good, everyone? 1 minute. I'm just closing it. So good, everyone? I'm stopping it now. All right, yeah. So out of all these, this is the correct one, which is the hemidesmosomes. Remember that adherens junction, desmosome, gap junctions, tight junctions, these are all on the lateral surface present as well. But the hemidesmosomes attaches the cells to the basement membrane. So let's take a look at this quickly just to have an idea where they are. See that these are the hemidesmosomes, which is attaching the cell to the basal lamina right here. On the other hand, you have these tight junctions, you have the gap junctions, you have the adherens junctions, and desmosomes all are found in between the cell or the lateral side of the cells. So that's about it. All right, very good. So let's move on to the next material. Okay. So next, what we are looking at is the second type. So that's all about your occluding junctions. Next, we are moving into the anchoring junctions. And cytoskeletal filaments are responsible for that. And this gives the structural integrity of the cell. And they are abundant in the heart muscle because the reason, think about that, that you are, for your whole lifetime, you are constantly beating your heart and the blood is being rushed through that. So it's a lot of mechanical stress. And also they are in the skin epithelial cells for the mechanical stress, as well as think about your nails and hair. The important part is that your top layer of your hair or skin, they are dead cells. But even those dead cells are still intact with these desmosome and desmosome structures. They are connected to each other, and that protein doesn't degrade that easily. They are present in that dead cell and keep the integrity of them, of your skin. As well, they are also, which is not mentioned, not needed, but they are also present in the blood vessel because blood is flowing constantly. They are present all through from your inside your mouth to all the way to the anal opening. There is all throughout the gastrointestinal tract. They do have these particular anchoring junctions because you have mechanical movement of material. They are present in the urethra. They are present in the ureter, which also flows the urine. And so anywhere you can see that there is a mechanical friction or there is something which flows on top of these cells, you will have these anchoring junctions in abundance. There is always there in other structures as well, but they are in abundance in those areas. So what are the two types? You have these adherence junctions. Adherence junctions are made up of actin, and we are going to take a look at these adhesion belts and focal contacts. Whereas the intermediate filaments, these make the desmosome and the hemidesmosomes, and these are actually structurally much more stronger compared to that actin. The actin is finer, and they are a little bit, think about that this is a little bit of, the diameter of these ropes are a little bit less compared to that. So that's the overview of that. That's basically what it is. So what we are seeing that in between the cell, you can see that these are the structures, which is in the black on the lateral side. These are some of the anchoring junctions. So this is in between the cell, and right here is your extracellular matrix. It is also attaching them in between them. So we will have an example from both actin and intermediate filaments. So all of these do have a basic similar structure. So what are these basic similar structure? So what you have, suppose this is your cell A or cell 1, and this is your cell 2 adjacent to that. Bottom, you have the extracellular matrix. In between the cell, you have this extracellular space or intercellular space, that white area. Now what you have right here, there is a transmembrane protein, and these transmembrane proteins are known as the transmembrane linker proteins. So you can see that there is a domain right here, this square, which is interacting with the domain of the adjacent cell. And then there is a membrane part which is traveling through into the cytosol. So that's your transmembrane part. So these proteins are on the lateral surface of these two cells. And there is a domain which interact with each other. The next part, what you have is what is called as intracellular attachment protein. So here you can see that in the blue part, there is an attachment protein which is attached to these transmembrane linker protein right here. And then what happens that this intracellular attachment protein, on the other hand, will be attached to a cytoskeletal filament. This right here is the red, is a cytoskeletal filament, which could be either actin or it could be an intermediate filament. It varies depending upon what it is. So that's basically the generalized structure of the anchoring junctions. Now, here, it is basically the same. What you are seeing right here, instead of this transmembrane linker protein, here the linker protein is inserted into the extracellular matrix, and it travels through the basal side of the plasma membrane. And you can see that right here, you have the transmembrane linker protein, just exactly similar to that, and that is attached to the cytoskeletal structure. So that's basically what it is. What the important part is that when you do see that these proteins right here, like this, or even right here next to these linker proteins, these intracellular attachment proteins, when these are present in the cell, they actually form a kind of a dense area, and we call these as a plaque. So these intracellular attachment proteins form these intracellular plaques as well. So we're going to take a look at these in a little bit more detail. So right here, first we are going to look at this adherence junction. And interestingly, what you are seeing, this is your apical surface. This is a cell is a kind of a sideways. Remember that it's not top bottom, sideways. So these are the microvilli. This is basolateral layer right here in between the cells. And you have these tight junctions. Below that, you have these adherence junctions. That's what we are going to see first. And how it is, you can easily see that right here, apical surface, and this is the basal surface, and you have these adherence junctions, which are a belt-like structure, and we call these as adhesion belts. And these are extremely thick, and in a 2D, it's very difficult to identify. This kind of gives you a 3D structure, but remember that this particular belt is going around the cell 360 degrees. The same thing that it was occluding junctions, it goes 360 degrees around the cell. It is not that it is only on one side and it is not on the front and the back. No, it's going all throughout. So that's your adhesion belt, really, really thick. And what you are going to see that here, you have these linker proteins that what we have seen in the previous picture right here, which is in green. These linker proteins are known as the E-cadherin, which is right here. These linker proteins, E-cadherin, which is interact. So the E-cadherin of this cell will interact with the E-cadherin of this cell in the extracellular space. Then what you have, the intracellular attachment protein are known as the catenin and the vinculin, which is inside this cell as well as inside this cell. And to these catenin and vinculin, the actins will be attached to those. Actins as well as myosin 2 filaments will be attached. And why it is actin and myosin 2 filaments? Remember that the actin is the cytoskeletal filament right here as a microfilament. Myosin 2, on the other hand, is a motor protein which walks on the actin. So we are going to see that these adhesion belts can get squeezed and relaxed. And we are going to have some particular idea about that how does that work. So that's also very important. So again, remember that how this works. So just make sure that you have this intracellular cadherins, E-cadherin, then you have the intercellular cadherin, then you have intracellular inside the cell catenin vinculin attached to the actins. And on the actin, you have these myosin 2 filaments which can move, and this whole adhesion belt can be squeezed as well. So I just repeated this concept. So huge application or consequence of that. Think about that. If you are looking at a blastocell being developed into an embryo, now you know this. So think about that this is actually the top portion of the blastocell or the blastocyst, and you have this sheet of epithelial cells. So right here, this green line is the adherence junction or the adhesion belt. So what happens that in a particular way, these myosins actually squeeze these actins. And if you carefully look at that, all these cells right here basically are tapered towards the top. Very carefully look at that. They are tapered towards the top. That is happening because of the tightening of the actin or adherence belt right here. Whereas if you see the cell in the corner right here, they are relaxed. And this allows this to invaginate into this structure to form this epithelial tube, which pinches off from the top layer. And this forms what is called as a neural tube. and this will form the brain and the spinal cord. So let's see. So right here, think about that this is basically the structure, and this portion right here is right here. And what is going to happen, that this portion that you are seeing, this will be pinched off like this from that top portion. So that's basically what the idea that you are looking at. So this is basically going to get away, and that's going to form that neural tube on the top of that. So from this epithelial cells, that's how this is formed, basically. All right, so that's your adherence belt. The next actin-related structure, this is very, very common, and I will not need to spend much time here at all, which is the focal contacts. And you can see that this cell, which can, if you are culturing this in a culture, this can be attached to the matrix, or it can move onto this matrix. And these are the focal point where you have these connections. And you can see that this whole orange area is very rich in actin. And these are the other interacting protein. So what they are, your transmembrane linker protein is known as the integrin, so which is sticking out through the plasma membrane. And the intracellular attachment protein is talin and alpha-actinin. And talin and alpha-actinin will attach to themselves to the actin. And this is basically the way the focal contacts work. And now this is the picture that I was talking about. Very, very simple. So as the cell moves, if it's a motile cell, constantly these focal adhesions are going to get attached, and they are going to get detached as well. So there is a regulation of that. So you can see that these are the focal contacts, and the lamellipodia will extend into this area. So the cell is moving towards the right side. You can see the new adhesion. This will touch down. And right here, you can see that this is the old adhesion, which is a de-adhesion. So here you can control these movement like this. So this is your focal contacts, and again, with the help of the actin and the related proteins, which is the integrin and talin and alpha-actinin. All right, so after that, this is the new part that we are talking about, the cell-to-cell anchoring junctions, and this is related to the intermediate filaments, and desmosome is the structure. So right here, you can see the desmosome, and you can see that these are the two plasma membranes separated, you have a little bit space, but you can see that there is a dark band just next inside that plasma membrane. This dark area is known as the plaque. Those are your intracellular proteins that we're going to see in a picture in a second. Here it's much more clear. So you can see that right here, this is your cytoplasmic plaque in the dark, and you can see that these are the intermediate filaments which are running through the desmosomes. And these are the plasma membranes right here, two adjacent cells. All right, so this is the overall structure. So you can see that right here, you have the two plasma membrane of two adjacent cells, one here, one here. And you have these proteins right here, and these are the transmembrane linker proteins, and the name of these linker proteins are desmoglein and desmocolin. These are the cadherin family of proteins as well. So we talked about the cadherin or the E-cadherin, but these are the same family, just have different names, similar types. So they move through the plasma membrane, or they are stretched across through the transmembrane domain into the cell. And that's your plaque protein or the intracellular attachment protein. And the name for those are the plakoglobin and desmoplakin. So here is some of the little bit of memorization of names, plakoglobin, desmoglobin. And to these plaque proteins or the intracellular attachment protein, you have these intermediate filaments which are attached. And it can also depend that what kind of intermediate filaments. One we talked about in the epithelial cells, this is keratin. Keratin is also all over your skin cells. This keratin, you already know that it is an abrasion resistant protein as well, but along with that, it also anchors these cells via desmosome. And it is called desmin in the heart muscle cells, which attach to each other. So that's your structure of your desmosome. So here is an autoimmune disorder, which is known as the pemphigus vulgaris, where you raise antibody against the desmoglein. Remember that the desmoglein is one of the transmembrane linker proteins. And in here, you raise antibody against that and mainly affect in the skin desmosome. So here you are seeing that this is a big blister on the scalp. And here you can see the back, lots of these blister. And due to this problem, because what happens that there is fluid accumulation in between these areas, and as the cells are cannot be connected together in a tight, so the fluid basically what happens seeps through into the epithelial cell layer from the bottom connective tissue layer and gives this characteristic blister. But interestingly, what happens that only in the skin this is much more prominent. So know the name of the autoimmune disorder. So the next what we are looking at is the hemidesmosome. So the hemidesmosomes, on the other hand, is a very similar structurally. So the only difference right here, the desmosomes are in between the cell, whereas the hemidesmosomes are connecting the cell with the basal membrane to the basal lamina or the basement membranes. And here you have this transmembrane linker proteins are integrin. So you can see that they are now different. They are different than the desmosomes. So they are not the same. They are integrins, whereas the attachment protein are desmoplakin-like proteins, but not exactly desmoplakin. And you can see that they are attached with this network of intermediate filaments. So that's your hemidesmosomes, and that's how you can anchor these cells together. All right, so here is a really good chart where you can see cell-to-cell adhesion belts, cell-to-cell focal contacts, both of them acting. And then you have transmembrane linker, catenin, vinculin, taline, alpha actinin. Here you have cadherin, and then you have ECM proteins, and then desmosome with the intermediate filaments, hemidesmosome with the intermediate filaments. So this is a good chart. You can keep it handy. It can really just make sure that you have this one, and you can consult this when you take the fourth exam. Will be a little bit helpful. But you do not have to know those basically where to apply these things, but it will just a little bit help in your memorization. All right, so the last one is your communicating junctions and the gap junctions. And we just touched about this in just one slide when we talked about the cell signaling. So what are the gap junctions? And you can see right here in just one picture, you have a tight junctions, you have an adherence junction, you have a desmosome, you have a gap junction. It's a beautiful electron micrograph. So right here, what you are seeing that there are two adjacent cells, and right here is a channel forming protein complex. And this is just a kind of a very crude way to distinguish that what is a gap junctions. Gap junctions allow some of the molecules to pass in between the cell, but not all, usually very small. So here you can see an electron micrograph, which cell 1, cell 2, right here, gap junctions. Here you are seeing the gap junctions if you see it from this side. So basically this pore that you are seeing going through, you are looking at the pore, down the pore, and that's why you can see that it's honeycomb-like structures, what you are seeing. So these are 90 degrees to each other. So you are seeing from the top cross section where you are seeing right through the pore. That's your gap junctions. So interestingly, what happens that you can do an experiment where you can microinject a fluorescent dye, or you can microinject a protein or a larger molecule attached with a fluorescent dye. You can just do this experiment very, very nicely. You can take a fluorescent molecule just by itself. Suppose the molecular weight is 100 Dalton for that. And that, if you inject into this cell, that molecule will pass. Then suppose about 1,000, you just take a dinucleotide attached with a fluorophore, and that will even pass through this, or a vitamin that will pass through. So on the other hand, if you can see that if you have a 5,000 Dalton, like a very small peptide, you attach a fluorophore to that, it will not pass. Any protein, absolutely not going to pass. So you can easily see that the gap junctions only allowed molecules which are approximately about 1,000 Dalton or 1 kilodalton, 1 kDa or 1,000 Dalton can cross the gap junction. So your 1,000 Dalton is 1 kilodalton, kDa. So what are these molecules? It could be inorganic ions, it could be amino acids, it could be nucleotides and vitamins. They can cross in between, but it will not allow the proteins, peptides, nucleic acid, polysaccharides cannot cross the junctions. No, so remember that even the viruses cannot pass through this. They are, because viruses are made up of, they are way larger. This is just only one protein. They are made up of many, many proteins. They cannot pass through this either. So for signaling that this helps. So you already know that the heart muscle, all the muscle cells need calcium. And the calcium will easily transport it from one heart muscle cell to the other. And these gap junctions are abundant in between the heart muscle cells. You will learn it from somewhere. If you take an anatomy physiology course, you will be able to see that. So let's take a look at that, how this works. What you are seeing right here, you have the left-hand side, you have cell 1 and you have a cell 2. Interestingly, each of these gap junctions are made up of a protein which is known as a connexin. This connexin is a 1, 2, 3, 4 pass transmembrane protein. So you can see 4-pass transmembrane protein, and these transmembrane protein, these connexin, they form a connexin with 6 of them. So you can see that this is one connexin right here, 1, 2, 3, 4, 5, 6. So 6 of these connexin, which is this, form this structure, this pore. So here in the pore, you have this 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So you have this, and interestingly, what happens, you have this transmembrane protein on the cell here, on this side. You have a corresponding transmembrane right here, and again, randomly. When they match up, they will form a continuous pore. So you have the connexin made up of connexin of this cell. You have a connexin made up of connexin of this cell. When they basically line up against each other, you have a continuous channel through which some of the small molecule can pass. And most of the time, these are signaling molecules. So that's your gap junctions. So interestingly, there is a regulatory mechanism for your gap junctions. And what you can do, if you have a high calcium level and low pH, this is closed. So think about that. Again, I'm not going into the physiology. If your heart muscle do have enough amount of calcium, you do not need that. If it is needed when you have a low calcium, it will open it up to get calcium. And the same thing happens with high and low pH. And this is basically that if there is an anaerobic respiration going on, you have a lactic acid deposited. When you have too much lactic acid, low pH, when you do not have too much lactic acid, then it is closed. If you have too much lactic acid, it will open up. Those are some of the examples which is not absolutely needed. Just remember that there is a mechanism by which the gap junctions can be closed or opened by conformational changes of these each individual connexin proteins. And what condition they close? They are closed in high calcium, low pH, and they can open in low calcium in the high pH. So these are just some of the consideration. All right, so last quiz question is, which of the following can pass through gap junctions? Polysaccharide, GFP, glucose, ribosome, viruses. All right, stopping it now. Good, everyone? I'm stopping it now. All right, so if you see that the answer-wise, I have C as 41%, and glucose, that is actually the correct answer. So I'm going to explain that thing, that because you have 41% glucose, that's still the highest, but you still have 24% in here, and I will just explain that in a second, that why it is so. So remember that the glucose is, of course, a small molecule. It's a monosaccharide, it's a sugar, and it is absolutely much, much less compared to your any other molecule. This is the smallest one, and it will pass. See now what are the examples? The polysaccharides are multiple sugars, and these are very large. Say, for example, starch. Starch will not be able to pass, or glycogen will not be able to pass. These are the polysaccharides, multiple sugars, not pass. GFP, where the confusion comes in here, remember that, and a couple of people answered that. It's a green fluorescence protein. So what is the difference? If we just take a look at that, let me close this first. If we just move on to, oops, sorry, right here. So what I said right here, that these are your fluorescent molecules can pass through. Fluorescent molecules are a fluorophore of this molecular weight, but remember that here the GFP is a green fluorescent protein. It's a protein molecule, and it's approximately, if you remember that that's not needed, but a protein will not pass, and it is about 2730 kilodalton in size. But even if you do not know that molecular weight, it's a green fluorescence protein. This is not a fluorophore. It is much, much larger, and it will not pass through. So it is not a fluorophore, which is a much smaller molecule, which can pass. Very different. Glucose, of course, will pass. Ribosome will not pass. Many, many different proteins and RNAs, so much, much larger. Viruses will not pass either, because think about that. Viruses are made up of multiple proteins, multiple or nucleic acid. And although if you just think about that right here, right here, we said nucleic acid cannot pass, proteins cannot be passed through that, and the virus is made up of all of those. So it's too large to pass. So glucose is the correct answer. All right, so that's pretty much it. Anyone, any questions? Let me see.